Welcome everybody. This is Mirza Umar. Today I'm going to be presenting the topic that I have selected for my paper writing, that is CD30 CAR T therapy in relapsed refractory Hodgkin's lymphoma. So that is the topic. It is a clinical trial that is published in 2020, 23rd of July in Clinical Oncology Journal that I'm going to be attaching at the end of the slide. So we all know that our immune system is responsible for making white blood cells. We have different kinds of white blood cells like neutrophils, eosinophils, basophils, and along with T lymphocytes and B lymphocytes. So that paper basically made and genetically modified T cells, and they name those T cells as CD30 CAR T cells. That basically they introduced a genetically modified version of chimeric antigen in those T cells. So we will be looking how this works, but let's, for the sake of purpose, have a quick overview how our immune system recognize viral and foreign antigens. So we have T cells, and T cells are always patrolling throughout our body. So if they find any foreign antigen or any viral antigen on the surface of membrane, then they will mount an immune response. So T cells recognize antigen presenting cells, and antigen presenting cells are macrophages. When they encounter any viral cell, they engulf those cells, and then they process those viral antigens or foreign antigens over the surface of the membrane, and then those cells are presented to T cells, and then T cells will bind to those cells, and then they trigger immune response in the form of release of pro-inflammatory cytokines. So that is the overview. Normally, T cells can only recognize those antigens that are presented with MHC class molecules. So MHC class 1 and class 2 are the molecular markers that process antigens and present to the T cells. So it's a basic background that you need to know before starting this presentation. So the table of contents, I have divided this presentation. First, I will be going through the background of the disease. In our case, it is Hodgkin's lymphoma. Then we will be having the study overview, which is the main thing. Uh, mm, what scientists have discovered in the study, what were the materials and methods used, and finally we will explain the results, and at the end, discussion and conclusion. How was the results monitored, what was the response, and what were the research gaps and side effects and limitations of the clinical study. So let's start with the lymphatic system. In order to understand the cancer of B cells, which is Hodgkin's lymphoma, let's quickly understand lymphatic system. So we know that we have cardiovascular system that is responsible for transporting oxygen through blood to all over the body cells, and then we have another system which is called lymphatic system. As you can see in this picture, those are the lymph ducts which carry lymph, and that lymph is the fluid that is retrieved from the interstitial space around the cells. So that fluid can contain proteins, microbes, abnormal cells like red blood cells, and that lymph basically flows through those ducts and go to the lymph nodes. So basically lymph nodes are the checkpoints where that lymph is monitored and screened, so making sure that the lymph is free with foreign antigens or microbes. So if they find any foreign antigens, then they will obviously mount an immune response. So Hodgkin lymphoma is the cancer of the B cells. The B cells become cancerous. B cells are born in the bone marrow, but they travel and reside in the lymph nodes. As you can see, the hallmark of the cancer is that it metastasizes. It means that it spread to other body organs. That is represented by the stages of lymphoma: stage 2, stage 3, and stage 4. Hodgkin's lymphoma, as you can see in this picture, the lymph node is really inflamed, enlarged. So if that is not treated, so that it will spread to other organs and cause other complications. So microscopically, Hodgkin's lymphoma is characterized by the presence of Reed-Sternberg cell, that basically have distinct owl-like appearance under microscope. So that basically confirms that this person is having Hodgkin's lymphoma. It is marked by swollen lymph nodes, fever, weight loss, and night sweats. So the standard therapy includes PET imaging, chemotherapy, drugs, and stem transplant as well. In this study, also the patients that have taken the clinical trial were already receiving those medications as mentioned here. But the problem was that the relapse rate was very high. So even they go along these therapies, but the cancer comes back again and again. So the study aim. This research paper covers a clinical trial by genetically modifying T cells, and it was done in two major hospitals, University of North Carolina and Baylor College of Medicine in Texas. So let's now quickly understand T cell activation before going to the main theme of the study. So T cells are responsible for immune reaction, and then those T cells are patrolling all over the body, and if they find any foreign antigen presented by MHC molecule, they will bind to those antigens. That is here shown in the picture by T cell receptor that is bound to the antigen presented by antigen presenting cells along with MHC molecule. And then we have co-stimulation as well, where the T cell is binding with another receptor over the surface of antigen presenting cells. Once T cell gets activated, they will go all over the body, and if they find any of those antigens, they will bind to those antigens and mount an immune response and destruct the cell, and that leads to programmed cell death, which is apoptosis. So now our main concern for this study is CAR T cell therapy. CAR T cell, also known as chimeric antigen receptor, is an engineered receptor combined from other receptor parts to modify and specifically recognize cancer cells. So the normal cells in our body, normal T cells, they are only limited to recognize only those antigens that are presented by MHC molecule. And most of the time what happens in cancer cells is that cancer cells escape, and also they are not processed with MHC molecule. So in that way, our immune system cannot recognize those antigens, those cancer markers, and that is a limitation. So chimeric antigen is making sure that it specifically recognizes cancer antigens, uh, and also those antigens that are not processed with MHC. So it has a really targeted approach. Okay, CAR T cells are now the really hot topic in clinical oncology. Many CAR T cells are now developing. Uh, there could be many CAR T cells specifically to which the target is. So, in case of CD30 CAR T cells, in our case, which bind to CD30 proteins found on Hodgkin's lymphoma cells. Now let's talk about the architecture and the molecular structure of CAR T cells. We have obviously outside the extracellular domain that is connected by the transmembrane domain, and in the inside of the cell we have a intracellular domain that spans all over into the membrane. Uh, what happens? What is the function of extracellular domain? It will bind to the cancer antigen. Here in this case, tumor cell. It will bind to the cancer antigen and connects and gives signals to intracellular domain, and then intracellular domain will release cytokines and enzymes to self-destruct the tumor cell. Here perforin, which kind of make pores in the tumor cell, and then granulozyme, which release lysozymes, and that will lead to the death of the cancer cell. So that is a CAR T cell architectural molecular structure. Here, this is made by me. I have tried to differentiate between normal T cells and CAR T cells. Here, as you can see, chimeric antigen drawn here that binds to the CD30 protein presented by B cells or the Hodgkin's lymphoma cell. Okay, what is the overall workflow? To do this, first we take the blood sample from the patient. Then we separate white blood cells, and then we insert CAR T and CAR T gene in those T cells, which is chimeric antigen receptor, and then we culture and produce millions of copies, and then they are introduced back into the patient, and we that is the final step where CAR T cell attack cancer cells. So how CAR T cells work? That is the lentivirus. Here you can see that is the vector that is used to deliver antigen DNA, modified DNA that is responsible for making chimeric antigen receptor in the T cells. Okay, now let's talk about materials and methods used in the study. 28 patients were enrolled at Baylor College of Medicine, and then we have 17 patients at UNC. The normal age was 35 to 17 years old. Okay, those patients had already been receiving the drugs and stem transplant, as I already mentioned. Main goal. Main goal was to find the best safe dose and check how the safe dose works, and also monitor side effects. Result criteria: complete response, it means that the cancer cell will be completely gone. Okay, partial response means some of the cells are cancerous and some are gone. Okay, safety and side effects control. Complete response was observed in 24% patients. There were mild symptoms, no neurotoxicity was reported, but there was a skin rash that was observed in 48% people, and it was also mild. It resolved naturally. As you can see here, this is the picture taken from the paper as well. This basically tells the biopsy of the skin, which shows the rash that is not caused by the cancer cells, but it is caused by the immune response in response to the CAR T cell therapy. Okay, overall response was good, 62%, complete remission 59%. So if I have to discuss the drawbacks of this study, so the first drawback was I mentioned in the paper that this clinical trial was really small. The and the sample size was really small, so you need to have a really bigger sample to know the satisfactory results, firstly. Second, manufacturing CAR T cells is really time consuming. Often it takes weeks to months, and it might not work for those patients that are critically ill and they are at the last stage of cancer. So that is one of the drawbacks. Another drawback is that making CAR T cells, as mentioned, is really time consuming. So now researchers are working on infusing the chimeric antigen through CRISPR-Cas9 mechanism into the cell, so that naturally the body T cells can make the CAR antigen by themselves to avoid the lengthy process in the lab. Okay, future direction. First is that CAR T therapy is effective in treating blood cancers, but it is not effective in treating solid tumors. Why? Because solid tumors have a dense mass around them. Also, they have poorly defined antigens. So the CAR T therapy is not effective in solid tumors. Also, solid tumors have really limited limited mechanisms, and also they can escape from the immune response really quickly. So now researchers are working obviously to treat solid tumors by this, and also they are identifying more than two antigens, which is called tri-targeted approach, so that the cancer won't be able to escape. So creating CAR T cells that can recognize two or three antigens at once at the same time. There is another paper that I had read, which basically tells the CAR T therapy along with interleukin 18 release. So that is a natural immune boosting chemical that was released by the CAR T cells in order to bind more strongly and cause cytotoxicity against cancer cells, and also making sure that cancer cells do not escape, and they've to cause a longer lasting impression. Okay, final takeaway: CAR T cell therapy is really promising. It's a really promising start. There have been many studies going on to treat cancer cells. Recently, I came across a post on LinkedIn in which the brain tumor was radically reduced in just days, and the therapy used to treat the brain tumor was CAR T therapy. So, long survival. It has new hope for relapsed refractory patients, but it is still still needs to work on solid tumors, and also the bigger sample size, bigger methodology and materials, so that we have satisfactory results. Also, in this study, it is mentioned that CAR T therapy, those T cells do not stay longer in the body, so they basically disappear really quickly. So that is also the major drawback that I came across in the paper. So that's why cancer can regrow again. Okay, if you have any questions, if you want to have access to read the paper, I can mail you the links. And if you want to know further about CAR T therapy, you can always ask questions over my mail. So that is all from my side. I would like to thank all my classmates and my instructor, Dr. Kim. Uh, and that is the last slide. And I hope I explained pretty well about all the mechanisms. Those are the references. That is the paper that was a main paper in which I have written my peer review, my article. Thank you again.